Folks, our next speaker is Dr. Anna Thonis, and she's going to talk about a new initiative that was launched last year in May, June. It's a long-term monitoring study of the eastern box turtle. And Anna, you all set? Okay. You got your pointer?
Okay.
And as soon as you hit go, the time will start.
Okay. Thank you, Mike. All right, everyone. Thank you for being here. As Mike said, my name is Anna. I actually completed my PhD in Stony Brook Ecology and Evolution Department, so I'm excited to be back. And today I'm going to be talking to you about the long-term monitoring project that we started just like not even a full year ago here in Long Island in New York. So as many of you know, we have eastern box turtles in our backyards, or in many of our backyards, hopefully, on Long Island and in the New York area, but the species actually has a pretty broad range. Within this range, we do have plenty of areas that are, urban hotspots, so higher impervious surface cover here means more urban, so you can see different cities based on that. But what you might not realize, so this is within the eastern box turtles range, is that Long Island actually represents the most urbanized part of the eastern box turtle range. And so if we look at Long Island, we go here from, I think there's a pointer. We can't really see, a little bit of lag. Again, from west to east, you get more urban. And so when I was designing this study, which Mike and Enrico both actually approached me 2 years ago, when I gave a talk at the Long Island Natural History Conference, and they said, hey, do you want to start a long-term monitoring project on eastern box turtles? And I said, okay. And so I said, but if we're going to do that, and we want to collect important information on the box turtle demography and natural history, but I also want to ask an interesting question. So how does that demography and natural history and other characteristics change with degree of urbanization surrounding these habitats? And so what you'll see here is we picked 4 sites. We picked the Staten Island Greenbelt, Alley Pond Park in Queens, a site in Upton, New York, and a site in Southampton, New York. And as you can see, again, these do vary in degree of urbanization. So survey details, these surveys took place between May and June of 2025, so not even a full year ago, which is aligning with peak turtle activity. Each location, so of those 4 locations, was surveyed 2 days for between 6 to 8 hours per day. And we had 5 to 19 human volunteers per survey. Very importantly, we were also accompanied by a wildlife detection dog named Newt and his wonderful handler, Dr. Christine Hoffman, who goes by Chris. And so importantly, one thing that's really relevant here is to understand that Newt was 11 times more efficient than a single human searcher, okay? So this is really underscoring the value of actually partnering with these wildlife detection dog teams when we're actually trying to survey cryptic species like eastern box turtles. So we see them, right, like kind of randomly sometimes. What if we actually want to systematically survey them? These dogs help a great deal. And so what I want you to look for here is the box turtle, and I want you to see if you can find it. And you might be able to. This one's actually not that hard. And so it's right there. And the reason I show you that is to emphasize that when you're surveying these turtles, it can be very easy to skim over their cryptic hiding spots like this. So another example, this one's actually harder, so good luck. But anyway, again, we're scanning, we're looking for these turtles, and they really are pretty hard to find. So this one actually, if you get the right angle, you might see the spotting there. But anyway, another example here, this is a video. We're gonna see if this buffers a little bit. This is Newt looking for a turtle. I do apologize, this video is kind of chaotically filmed. I didn't really anticipate him actually finding the turtle in this video. So it's just kind of a random video that worked out. One thing that his handler Chris told me is that when he has the scent of a turtle, his tail will wag faster from left to right, as it is doing in this scene. And one thing that's also really important to note that what you're gonna see is when he finds a turtle, he's going to sit down and he's going to essentially just stare at us to say, hey, I found the turtle. And this is really noteworthy because some wildlife detection dogs are actually trained to pick up the turtle, but that can actually transmit viruses or diseases within the dog's saliva if the dog were to pick up multiple turtles. And so there he is. And so this turtle, you might be like, okay, well, you guys could have spotted that. It's literally right there. And like, yeah, you're right, that maybe we could have spotted this turtle. But what I'm going to tell you is that we could not have spotted this turtle. So this is a log, and this is the hole where Newt found this turtle. And this is a video that I tried to take with my flash on my phone to try to get you to see where the turtle was, and Newt found that turtle. So highly, highly effective in finding these turtles. So when we would find a turtle, we would collect things like straight carapace width and length, straight or plastron length and width, body mass, also the sex and the age of the turtle, as well as any signs of damage or disease. We would also take a blood sample from each turtle for downstream genetic analyses, as well as environmental data like wind speed, humidity, temperature. We even had a pollution meter with us so we could try to gauge the pollution levels at those sites because, again, some of them are more urban compared to less. We also, notably, gave each turtle a unique notch code. So this is how the notch codes look on these turtles. So you have 3 notches. So this turtle's notch code would be 1L, 8L, 8R. These notch codes are administered by the Department of Environmental Conservation per county. So we get these from the state, and then we give them these notch codes so that in subsequent seasons, like this coming season, if we find a turtle, we can tell if it's already been captured, and that way we can actually look at how much it's grown, or the mass has changed, or what has happened to this turtle. Maybe it has new signs of damage or injury. So getting into some results here. So year 1, we found 8 turtles in Upton, New York. I will note that these 2 days we were in Upton, it was definitely a bit rainier, which sometimes rain means more movement for the turtles, but what can also happen is that the rain, active rain, can wash away the scent of the turtle, which can make it harder for the dog to actually find the turtle. So again, pros and cons. However, also the Upton, New York understory is very dense, so it's very hard for humans, especially in that location. We found 13 turtles in the Staten Island Greenbelt, which was really exciting because this Greenbelt area is all built on a landfill, and so these turtles were reintroduced there in about the 80s, and there hasn't been a lot of resurveying efforts in that area, and we found a lot of new hatchlings and juveniles, so it's clearly a pretty healthy population, so I'm really excited to go back to that site again this May. Southampton was really overwhelming because why did we find 52 turtles? And so I was hunched over doing data all day, and the dog was going crazy, and the people were also finding them because the habitat here was very grassy, meadow-like, but absolutely beautiful, really exciting to see that many turtles. And then in Alley Pond Park in Queens, we actually didn't find any. And this is only notable because there used to be a known eastern box turtle population in that area. However, we had 15 volunteers and 18 another day in those sites, as well as the dog. We surveyed for about 8 hours per day. We found a Florida cooter, which is a non-native, is a pet trade, someone dumped it. We found things, we just didn't find box turtles. So I'm not gonna say it means there's no viable population left there for sure, but it doesn't bode well. So these are just our numbers if you want to see sex ratios. I could have probably made this prettier, and I do apologize, but the notable thing here is that, especially for Southampton, we literally had 21 males, 21 females, so it's pretty even sex ratios. Obviously, for the other 2 sites, I would like to get bigger sample sizes before I make any drastic claims about sex ratio. And then relating to blood samples, we did get 34 blood samples across these 3 sites, and so this season we'll collect more, and hopefully down the line we'll be able to actually say something about the genetic diversity of those populations, because higher genetic diversity will typically indicate, higher genetic variation, which will give way to longer hopeful population viability. So these are I'm gonna go through these kind of quickly. Body condition, it's kind of a proxy for fat stores in the turtles. There's equations that have been, used to calculate these things. Again, with Upton and Staten Island, I would like to have bigger sample sizes before we actually make any claims about the body condition, but what you can see here is that kind of across the board, there's not too much variation. I wanna definitely add in more sites before we look at this more. And then I also want to show damage and injury. So I think the most notable thing here is that Staten Island does appear to have the highest rates of damage and injury, and the reason I think that is notable is because this is theoretically one of the more urban sites. Queens and Staten Island were the more urban sites. I don't even necessarily know how much has to do with the urbanization, but there are more raccoons on Staten Island, and raccoons do love to gnaw on box turtles, and so we did see a lot of gnawing. You can see the marks. And so I could get into these in more depth, but I'm not going to just for sake of time. So this upcoming field season, we're going to be doing these surveys in May. I'm going to be resurveying these 3 successful sites, and we're also going to be adding a site on the border of Queens and Brooklyn, pending permits. And pending funding, we're hopefully going to be adding 1 to 2 sites on the North Fork in collaboration with the group for the East End, but again, that is pending funding. Additionally, this season we are going to be collecting toenail samples from each turtle that we catch. And this seems maybe a little strange. I'm not going to go into it too much, but it allows us to do things like dietary analyses, comparative analyses, looking at the trophic level of what these species are eating, as well as contaminant analyses. So like, for instance, do certain populations have more exposure to mercury and things like that? So that could be really interesting from the urbanization standpoint as well. And then lastly, new for 2026 is we are going to be joined as well by Canine April. And so she is only about a year old, or not even a year old now, but she has been very thoroughly trained in leading up to this season. And so we'll have 2 dogs, which hopefully means maybe 22 times more efficient, who knows, than a single human searcher. And so anyway, I would like to thank my partners on this, as well as my lab at NYU, Chris and Newt. And if you have any questions, please feel free to reach out. Thank you.